Hello, everyone, and welcome to Critical Path Institute's Rare and Orphan Disease Program Professional Development Webinar Series. My name is Dominique Cruz, and I'm a project manager for the Rare Disease Cures Accelerator Data and Analytics Platform, or RDCA-DAP for short. RDCA-DAP provides a centralized and standardized infrastructure to support and speed up rare disease characterization with the goal of accelerating therapy development for rare diseases. It is my pleasure to welcome everyone to today's webinar, Advanced In Vitro Modeling of Neuromuscular Diseases and Therapeutics. Before we begin, I'd like to go over a few housekeeping items. If you have any questions throughout the presentation for our speaker or our panelists, please put them in the Q&A box. The chat function is disabled, so we can make sure that we don't miss any questions that pop up. And all participant lines are muted so we can reduce background noise. We are recording this presentation and it will be available for on-demand viewing on our YouTube channel shortly after the webinar concludes. And we also have our contact information. If there are any questions that we don't happen to get to, please feel free to reach out to us at rdcadap@cpath.org. Now I am pleased to hand it over to Dr. Ramona Belfiore-Oshon, who will introduce our speaker today.
Thank you, Dominic, and good morning, everyone. Good afternoon, depending where you are. It is my honor today to introduce our guest speaker for today on advanced in vitro modeling of neuromuscular diseases and therapeutics. Dr. Francesco Saverio Tedesco is a professor at UCL, senior group leader at Crick, and a honorary consultant at Gosh. And with his large expertise in neuromuscular diseases and in vitro complex system, today will give us an overview of his work and progress. And thank you so much for joining us. I will hand it over to Saverio whenever you're ready.
Thank you very much, Ramon, for the nice introduction and invitation. Let me share my screen. Let's see if I manage to do it properly. Share. Can you see my screen? Any luck?
Yes, we can now. Thank you.
And hopefully it's in full screen mode.
It is. Good to go.
Thanks very much. So I will give you a brief overview of what my lab does and what is my main interest when I'm not in the hospital. I'm a pediatric neurologist by clinical training and a stem cell biologist to some extent, I probably should say, for the rest of my life and career. And I've always had a keen interest in trying to harness the potential of different type of human muscle stem cells for a number of applications, initially for ex vivo gene therapy, cell therapy, and more recently we focused quite a lot on using those systems to really try to recapitulate with as much fidelity as we can features of skeletal muscle, so that we can use that for developing potential therapeutics, or at least accelerating development for therapies still in need of a cure. So as I said already, I will be discussing and talking a lot about muscle. Most of you might be familiar with this, and muscle, it is the most abundant human tissue and is the main output of our central nervous system, so a rather important tissue and organ of our body. It has a complex structure and functions, and one example of that in terms of complexity is the presence of multinucleated myofibers. So you see an example here. Each muscle fiber, which is this purple bit here, forget about this red myofibrils here, contains several different nuclei inside. And this and the nuclei, obviously, is where the genetic information is stored. And this is particularly complex and challenging when we try to develop therapies that are, for example, genetic therapies that need to target every single one of these nuclei, or at least the majority of them. And these muscle cells can be remarkably large. Some muscle fibers can be centimeters long at variance with other cells, which are much, much smaller. And unfortunately, muscle is the target of a number of different severe and incurable conditions with gene changes, with mutations really impacting on different parts of the muscle tissue, different compartments of this tissue, from the envelope of the nuclei, this membrane that surrounds the nuclei, as you will see later on, to the sarcomers, which are the contractile protein, the bits of the fibers which confer the possibility to generate force, and the muscle membrane as well, and the extracellular matrix, so the matrix surrounding those fibers. So and that tells you how big the problem we're facing is. And unfortunately, most of these conditions are incurable. And therefore, for a lot of them, there is no clear mechanism as well. So we really need good systems to study them so that we can facilitate development of therapies. And a way to do this, and a big sort of effort and investment of my lab, has been to try to model human muscle diseases as better as we can in a dish in vitro. This is due to a number of reasons, particularly for after many years, both myself and a number of other groups try to do complex studies like this in mouse models with some limitations. We decided also to focus on systems which will be much more relevant to some extent, where the research is started in humans in the first place. And we want these models to be patient-specific, time-resolved, and scalable, and to give us the possibility to dissect molecular pathways. And a solution to do so is really to try to make muscle from human pluripotent stem cells. Now, I would assume that most of you are familiar with the concept of human pluripotent stem cells, which were at the basis of the Nobel Prize for Shinya Yamanaka over a decade ago now. The rationale there, the logic, is that you can take a somatic cell, which is any cell type of our body, really like a skin cell, turn this cell into a pluripotent stem cell, like an embryonic stem cell, with a bunch of specific factors. And now you have a cell which is capable to differentiate in any cell type you might want it to become, without having to rely on an embryo as the original source. Back in the days, I was lucky enough to be in the right place at the right time, to be one of the first to take human fibroblasts from patients with different forms of muscular dystrophy, turn them into pluripotent stem cells, and then differentiate them into muscle-making cells and then myofibers, which were also then genetically corrected and transplantable. Now, there are different ways to do that. It can be done with or without forcing the expression of an exogenous gene, or in other more elegant ways to mimic the developmental stages that the embryos have to go through to develop muscle using specific small molecules. And this is just an example from our old paper here, showing that we can make lots of muscle. All this red stuff you see here is myosin, which is an important protein of those sarcomers that help muscle contract. You see there's a lot of it, and you see the multinucleation that I was referring to earlier. But you can also appreciate from this image here that the architecture, the organization of the tissue is not as you would expect it to be if you know a bit about muscle. You see these fibers going pretty much in every direction. And I will come back to this point and why this is relevant. I was pleased to see that then, following that work and the work, also seminal work of the lab of Rita Perlinghero and Jacques Tremblay and others back then, there has been an explosion of other platforms and systems to develop similar strategies to differentiate muscle from iPS cells. So now, depending on what you want and what you need to achieve, you can pick your favorite protocol and facilitate that aspect. But I mentioned briefly about the architecture, the structure of the muscle. Is it really 2D, like a monolayer of cells, enough? Muscles is pretty complex. It's a very three-dimensional tissue. You got tendons pulling the tissue from one hand to the other, conferring both passive and active tension to the tissue. You see this cross section here. If you slice the muscle horizontally, like this, for example, in your bicep or in one of the other muscles, you see this this nice arrangement of the myofibers and this three-dimensional extracellular matrix all around it. So we really probably need something better than just a monolayer of cells to recapitulate that complexity in vivo. And I will cut a long story short and avoid showing you some of the rationale to get to this point. But over the past decade, we did also a lot of work to move from 2D to 3D, really, to take those iPS and other human stem cell systems from a single monolayer to a three-dimensional structure, which not only contains myofibers, as you can see here in this branch of this cartoon, but also other cell types like neuronal progenitors, motor neurons, pericytes, endothelial cells. These are all like important supporting cell types to give innervation and vascularization to the cells. We combine them with biomaterials. We tweak systems that were previously developed also in collaboration with Thomas Eschenhagen to make cardiac muscle, and we basically developed what was a human fully iPSC derived skeletal muscle, engineered skeletal muscle that was also capable to model features of muscular dystrophy. I will show you more of that in a minute. And then more recently, last year, we also expanded the characterization and the applications and the remits of this platform to other systems, as you will see also today, particularly around gene therapy. And then obviously we're not the only one working on this, but we proud to have been one of the very first to start putting our hands into this very booming field nowadays. And now that you will see different groups and different excellent academic and industrial labs having their own platforms or variations of the team to get to these or similar systems. And again, these are just some examples, not much up to date really. This, and you can tell already how many options we had already 3 years ago. Now there are even more options depending on what you want to model, which tissue compartment you want to model, to look at your specific protocol to get to a three-dimensional engineered muscle for your research. This video here, just to give you a rough idea of how those engineered muscles look like if you image and stain the outer layer of the fibers. This is about 1 centimeter long, and you see now the myofibers running from end to end, stained with titin, which is an important protein of the contractile protein of the tissue. And for the sake of time, I won't be going much more in details around the dystrophin, which is not very key for this staining. Obviously, muscle contracts, and we want to have this piece of artificial or engineered tissue to do the same. And you can see that nicely upon electrical stimulation. And this was great work set up in the lab by Valentina Nonello, a very talented postdoc. You can see that upon electrical stimulation, they twitch nicely. And this is plotted here in this graph. And you can also, during those stimulation, image, and this is a bit faint, but hopefully you see it, these calcium spikes, flashes, telling you that there is so-called excitation contraction coupling. So the excitation stimulus triggers calcium release, and which eventually results into contraction of the tissue, telling us, to cut another long story short, that we have a functional piece of muscle that we can work with in the dish in the lab. And importantly, I mentioned this briefly in the cartoon, but now I'm going to show some of the images. We have also the possibility to engineer these muscles to contain not only myofibers, as I said, but also motor neurons. And this is work that we initially did with Ricky Patani here at UCL and the Crick, and then expanded also to other protocols. You see here an example of some of the motor neurons here that we incorporated in our tissue. And obviously, you know, we have a model here. This is by no means something which recapitulates with high fidelity. the aspect and the structure of a mature piece of muscle. But as you will see in the next few slides, we have a piece of tissue which is good enough to give us some information on some specific disorders. And here in this example, you see some motor neurons in green, the myofibers in gray, and some of the vascular cells, the blood vessel cells in green, in red or in yellow. And those muscles and motor neurons can touch each other and form these neuromuscular junctions here. Now, we have a platform, we have a system. Can we do something useful for neuromuscular disorders, which is our interest in the lab and my also interest as a pediatric neurologist in the clinic. So the first target disease we started to work on in close collaboration with my friend and colleague Pete Zammit at King's College is a group of disorder caused by mutations, by gene changes into a gene called LMNA, which codes for a protein called lamin AC, which essentially lives here underneath the nuclear envelope. So it wraps around the cell nuclei, conferring structural stability and regulating gene expression. And when dysfunctional, it gives rise to this group of conditions called laminopathies. You might have heard of the progeria, the premature aging syndrome, which is also a laminopathy caused by mutations in the same gene. But the largest group of laminopathies really is the one affecting striated muscle, so both cardiac and skeletal muscle. And amongst them, the most severe is this condition called LMNA-related congenital muscular dystrophy, LCMD. There are other muscular dystrophies or muscle disorders caused by dysfunctional lamin AC, LMNA. But LCMD is by far the most severe, and in some very severe cases, you can tell already from these pictures here, from these photographs, that the children are unfortunately very severely affected with prominent contractures requiring also assisted ventilation, invasive ventilation, or feeding support, and unfortunately with a short lifespan. Despite that, still, as of today, there is not much known in terms of genotype-phenotype correlation and pathophysiology in general, disease mechanism in general. So we decided to embark in this project using our platform to answer some of those questions and also to develop therapies for this condition, which remains incurable. And again, to cut another long story short, because a lot of this work is published, we show that when we took iPS cells from different patients with LCMD with that very severe condition, and when we turn them into muscle making cells and we make engineered muscles out of them, we show that when we would put them in 3D, the aspect, the shape of the cell nuclei, which you see now here in these two images in red, this is the cell nuclei where the DNA is stored. You see in the healthy donor fibers, which you see are running here in this direction and in this direction of the engineered muscle, the nuclei look pretty much unremarkable, like oval or rounded as they should. But when you look at the same into an engineered muscle with a mutation in the LMNA gene, so an LCMD muscle, then you see already here that the nuclear shape is severely dysmorphic. They are particularly elongated in this example. And this video shows you the sort of same information in a three-dimensional fashion. And this is quantifiable. You see an example here. And what is relevant, and as you can appreciate here, is that when you will do the same exercise of differentiating those iPS cells in 3D, so in the engineered muscle, you will see a higher amount of nuclear abnormalities, suggesting that we have a better way to recapitulate that feature in an in vitro system. And we also show that those abnormalities correlate with disease severity. So, for example, here you see this is R249W, is known to be the most severe mutation, the mutation giving rise to the most severe phenotype. And indeed, here is where we have also the highest number of nuclear abnormalities. We characterize in detail this nuclear elongation as a disease-associated phenotypic readout, and colleagues in France independently also show similar results in a different engineer system for the same condition. So sort of telling us that we were in the right direction as well. Now, this is an example of a few, like 4 or 5 different conditions that we are currently working on in terms of understanding the disease mechanism using a system that wasn't really possible in like a decade ago. And another reason why this is also relevant in this particular condition is because the animal model has some limitation. So this is a so-called autosomal dominant condition. So there is a gain of function mechanism causing the problem in humans, whereas to see similar problems in mice, we need to have a loss of function. We need to knock out both of the alleles. We need no LMNA, no lamina C at all. So telling us that the mechanism is in mouse is different, and therefore using a human system might be to some extent also more informative. But what are we doing in terms of therapies? Let me give you another example now from the therapy angle. We working particularly around advanced therapy medicinal products, ATMPs, of which gene therapy is probably one of the things we are investing the most at the moment in terms of using this platform. And I'm going to show you also an example around cell therapy as well. I'm not going to be telling you much about tissue engineering, which is another important branch of ATMPs, but we show already some of this in a paper a few years ago. So gene therapy. Why do we want to use this platform to optimize gene therapies? Well, some of you might be aware that the neuromuscular field has been lagging behind for several years in terms of gene therapies, and then now we're going particularly fast, actually, and we've had one of the most successful drugs, like Zolgensma for spinal muscular atrophy, with impressive clinical results. But at the same time, we have a number of examples of different muscular dystrophies and neuromuscular disorders in which adeno-associated viral vector gene therapies, AAV-based gene therapies, has not been so successful for different reasons in terms of specificity, but particularly for toxicity and particularly, for example, for liver or cardiac toxicity. Just thinking about examples in myopathies like in X-linked multiple myopathy or Duchenne muscular dystrophy, where we have exciting, promising results, but we're still not getting to the same level of efficacy in some cases. For example, in the case of Duchenne, or we're still dealing with toxicity problems when it comes to multiple myopathy. So for this and other reasons, we decided to focus on that question using our platform, and also to try to understand a bit better about what will be really the specificity for the different human tissues of the various AAV serotypes that are around, for which most of the knowledge has been gained in either rodent studies or dog or non-human primates, but very little is known in terms of the tropism against human tissues, rather than what we then understand after clinical use. And to answer this question, we had the pleasure to lead a large consortium which we have established recently and which started last year, called MAGIC. This consortium puts together a number of academic, private and private partners and patient advocacy groups from different fields and different expertise: immunologists, vectorologists, virologists, gene therapy expert, neuromuscular clinicians, and so on and so forth, to really tackle this question: can we develop more accurate, predictive in vitro models of neuromuscular disorders, which will then in turn facilitate human-specific development or testing of a number of different viral vectors for neuromuscular conditions. And this is obviously a very ambitious question, and we are now 1 year and a bit in the project. So we are starting to get some exciting results, some of which I unfortunately cannot talk about today for a number of reasons, but I want to give you a flavor of the idea behind this. And this is just a recent example, pretty obvious to some extent, but for us, quite important to have the initial proof of principle for this sort of consortium some time ago, in which you can see that we can easily, in one of these engineered muscle systems, have a straightforward dose response situation in which we have low dose and high dose AAV9, which is obviously one of the AAVs in current clinical use. And here we're just looking at the expression pattern of a fluorescent protein, and you can see when you put a higher dose over time, you can monitor in real time in a human piece of muscle how much of your transgene is going to get expressed in your tissue of interest, in this case muscle, which is not something easily doable otherwise in humans. And this is obviously quantifiable, and we have now obviously lot more work that we are developing in this consortium following this approach. And another example I want to give you also from our consortium is work that we're doing together with my other friend and colleague Mario Amendola in France at INSERM and Genethon. So with Mario, we are tackling another problem, which is actually use our engineered muscle platform to test gene editing, so so-called gene surgery strategies for, in this case, for Duchenne muscular dystrophy. So what we are working on and what Mario devised in the first place is a way really to unleash again the expression of utrophin, which is a sister protein of dystrophin, which is the protein absent or reduced or dysfunctional, if you wish, in Duchenne muscular dystrophy. And utrophin it can sort of do the same job of dystrophin, but unfortunately, after birth, it gets significantly shut down in our muscles and is only expressed in specific parts of it. So with Mario, we've been testing the hypothesis that using our engineered platform system, we can test whether reactivation of this utrophin protein by de-repressing, so basically we are removing a stop signal from this expression, can help potentially, can have a positive effect in rescuing some of the functions of these dystrophic muscles. And here you see in this example that we treated with his approach some of the engineered muscles, and you see now expression again of utrophin in the periphery of these muscle fibers. Here you see around the muscle fiber there. And you see this is quantified over there. But more importantly, if you then want to look at more closely on how are these muscles performing, and you can see that now the muscles that have been engineered to re-express utrophin are having a better handling of calcium, which, as I showed you before, is important for excitation contraction coupling, and they contract more often. And we also have more recent data that I haven't had the time to show you that they also contract better and stronger than the controls. Finally, and I'm almost done, another quick example of another ambitious approach of advanced therapy medicinal product for muscle, which is cell therapy. Cell therapy has been kind of a holy grail of the field that has been elusive for a number of decades now, due to a number of complexities, including the limited fidelity that we have from experiments done in mice when it comes to translating those findings into humans. And what Sang Choi has developed in the lab is a platform to really mimic muscle stem cell transplantation in an engineered platform. So a sort of an in vitro transplantation system to see whether we can get any readout of efficacy of the transplanted cells. And here you have an example where muscle, this is the sort of pipeline where we have engineered those muscles, we induce an injury using a snake venom, cardiotoxin here, and then we transplant human iPS cell-derived muscle cells, and we can then monitor how they go around and differentiate and migrate and contribute to the tissue over time. I'm going to just show you an example on migration. I won't have the time to show you more data around engraftment, differentiation, and function, which is work that we are finishing as I speak. But this is just to give you an example. In this video, you see these little green cells moving around on top of these injured white myofibers. And this gives you an idea how you can model something otherwise impossible to do it in another system, which is human cell transplantation in an isogenic fashion. So somebody's muscle stem cells transplanted in their muscle, and then you can look how well they can move around. And we showed in this work that we can predict the effect of some pro-migratory molecules such as this DLL4 and PDGFBB on the treated cells in the same way we did a few years ago in this work, in which we had to do the same pipeline, the same work in animal models, suggesting that we can do that in a fully humanized setup in vitro. And with this, I have finished. I close with a sort of an important quote I do every time, which I think is more important than take a message here, which is that all models are unfortunately wrong, but some of them are useful, and hopefully some of this that I showed you today will be even more useful in years to come. And this is my acknowledgement slide. Obviously I'm super thankful to the entire lab, and they're doing all the hard work, and I'm getting the credit for it. But I showed you some of the data produced by Sumit, Dusty Dar, Seung-Woo Choi, Kati Jiang, Valentina Lionello, and Daniel Moore. I haven't had the time to show you data from other lab members, and I'm very grateful to previous lab members like Sara Maffiletti, Luca Pinton, Heather Steele, and also Shilpita Sarkar, who's now a collaborator from AstraZeneca. I've acknowledged some of our collaborators during my talk. I reiterate our work with Pete and with the colleagues at the Institute of Biology with Francesco Montoni, Maria Mendola, and also Andrea Serio. And these are our funders, and thanks again for your attention.
Thank you so much, Saverio. This was a tremendous summary, a large body of work, and we really appreciate this summary. I think this is really relevant and informative for many muscular dystrophies and rare specifically dystrophies, but also it really tells us more of these applications. Now going to introduce our amazing panel today. We are joined by Dr. Sander Eagle, the head of translational models and genomics and informatics at Biogen, Dr. Graham Marsh, the scientific director of CPAT Predictive Safety Testing Consortium at CPAT, and Dr. Nagisa Sardrian, the senior advisor for new approach methodologies at OIND and the FDA. Thank you everyone for joining us today. While we wait for some questions to go through the chat, want to advise the audience to suggest any questions you might have. I know that Saverio has proposed a great summary of what has been developed so far, and I would like to start with a question to Dr. Ingle to provide us some perspective on what are the primary challenges of adopting such technologies across industries in your experience.
Yeah, that's a great question. Certainly, it was lovely work, Saverio, that you presented. Those are the kinds of models that we all aspire to. I think the challenges have been many and varied over the course of development of these types of models. In the early days, it was really trying to find, appropriate cell sources that differentiated to a reasonably mature state that you could really say that they represented the right cell choice. And then not every lab has the ability to manufacture their own, chips and models. And, that for many industry partners is a real challenge because we don't necessarily have all the resources in one place to build our own models. Over time, those things have started to resolve themselves. We now have commercial suppliers of cell sources that are reasonably useful. And there are more manufacturers who are making off-the-shelf chips and sources that are useful. And now we've run into the challenge of, reproducibility of systems, making sure that when we put everything together, that we can generate the same data time and time again. And another one that's been really significant for us is actually scalability. To do a good experiment, we all know that you need to be able to have, positive and negative controls on your plate, as well as multiple replicates in the samples that you're trying to study. And that is often a huge challenge with systems that are available, because they may be able to handle, sometimes at the very low end, 1 or 2 samples on a chip. Now we're starting to see ones in which, there's a reasonable number, maybe 24 or so, but we really do need to get to the place of 96 or, even more, because oftentimes, while we'd like to believe that drug discovery is a really elegant activity, in point of fact, it is oftentimes a bit of a brute force activity where we need to test a lot of things in order to really understand what we're working with. And so those are just some of the challenges that we're faced when we want to bring in these really elegant models. And then, of course, there's things like having a skilled workforce, and then the cost and time that it takes to develop these and really implement them. And that too can be a barrier to some of the use of some of these models.
Interesting. Thank you, Sandy. Yeah, that's absolutely something we will consider. And I appreciate your feedback on this one and given your expertise for those applications. I want to go to Dr. Sandria, and I know we have discussed many applications of this and the participation within the Critical Path Institute and some of those discussions related to the context of use are very important. Could you tell us a little bit more about what are the efforts that the agencies have in place to support the development of these in vitro models?
Yes, I can talk a little bit about that. There are a number of different activities going on right now, but they are, I would say, mostly focused on training reviewers to develop the necessary skills in interpreting data and becoming familiar with the types of models that are being developed and that are closer to maturity, I guess, in certain ways. There are research efforts that are ongoing, but again, the research efforts are really mostly to try and understand the variability amongst platforms. The platforms are so diverse, and what we've identified is that what a manufacturer of a platform kind of says that what are the conditions and how they characterize it. Trying to recharacterize it is not actually, doesn't work. When you try to reproduce the work, it doesn't, you can't replicate their work that easily. So because the systems are so complex and some of the supplies are so specific that recharacterization becomes an issue. And so we are trying to get some level of experience with these platforms to have some more comfort with the data and confidence. But I would say that we're working with we're engaging with stakeholders, both industry as well as manufacturers of these platforms, in trying to do a number of different things in order to make sure that we start kind of moving closer. They move closer to our thinking and we move closer to their thinking a little bit. One of the things that's important to, I think, people to understand is that I think there's not that much of a, or there's sort of a little misunderstanding about what the regulatory process actually is when it comes to these types of complex models. For one thing, we're not getting a lot of this data submitted to the agency right now. A lot of the models are being used for discovery, and that data is not submitted to the FDA. They usually send us development data, which is what's needed in order to make regulatory decisions about the safety of the drug, and then the efficacy in clinical trials. And so for these types of models specifically, I think that that's going to be a challenge because I can't see that these models are going to be useful in safety assessment. I don't think that they're going to be replacing any kind of animal studies that are being done that establish, what the starting dose is in human clinical trials and other types of things. But I think that they are helpful in filling in certain gaps. Certainly they can help explain mechanism of action, mechanisms of toxicity, maybe if they can mimic disease progression and response to drug. There are cases, obviously, in like, for rare diseases or confirmatory evidence where this type of data could be useful. And I think that we're trying to identify some of these things and see how we can, work towards them. But I think that, what Dr. Engel mentioned, those are very important points to consider, that some of the main challenges are going to be technical challenges. How do you characterize the cells? How do you qualify cells? How do you make sure that you have enough different types of cell types that are going to be relevant for the specific disease? How many donors do you have to actually have? I think that when it comes to regulatory decision making, those are the things that are going to become relevant. And then what kind of reference drugs you use as your positive and negative control in the assay. So I think that these factors are going to be the things that when it comes time to coming to a regulatory agency, that a sponsor is going to have to demonstrate and also show what kind of considerations were important in the validation of their methodology. Because at the end of the day, FDA wants to be able to say, okay, we looked at this, they justified this model using these parameters. These endpoints are acceptable to draw this kind of conclusion. Therefore, we can use this in, assessing the safety or efficacy or whatever it is, to reach some sort of regulatory decision. But I don't want to discourage anybody from submitting this type of data to the agency, because even if it's not used in a regulatory decision making scenario, it will certainly educate reviewers. People will look at it and gain experience. So I just wanted to kind of mention that too. So I don't know. I hope I addressed the question.
Absolutely. Thank you, Nikki. So I think that's very informative. Also the perspective of really utilizing these methodologies for purpose of providing the totality of evidence that might support a certain process. So I think that's very relevant. Graham, I think we had some discussions on the importance of the pre-competitive work. This is, of course, a very high interest for a critical path. Could you provide us your perspective on how the predictive safety testing consortium is looking at this, and what are the efforts currently ongoing?
Yeah, thanks, Ramona. Definitely the work that we're doing in PSTC to bring together regulators and academics and model developers in the pharmaceutical industry to look for synergies where we can build these models to be more useful for regulatory applications. I mean, the beautiful data that Dr. Tedesco just showed has a lot of applications for showing efficacy of drugs that could be enabling for IND decisions at the FDA. And really, it's just a matter of how do we package that data? How do we show that it's reliable and reproducible in such a way that it can enable a regulatory decision? And that's the work that we are trying to enable in PSTC to advance these discussions and look for context of use where these models can help regulators show efficacy or safety of new drugs that are being developed for these rare diseases.
Interesting. Thank you, Graham. So this brings us really on also the importance of providing information and how do we translate the information from preclinical to the clinical studies. And Saverio, I think one of the things I would like you to highlight, what are the actual specific key advantages that you see in the utilization of these tools when compared to traditional models, or maybe just as a support of those traditional models?
Thank you, Ramona. So I think there are multiple good arguments around that. I mean, first of all, I believe that coming back to one of the points that was made before around scalability and reproducibility, that could be a challenge, but at the same time, it could also be an advantage here, because once we fix those problems, then we have systems which are much cheaper than an animal model to reproduce across different labs and scale up or down when needed. I mean, I haven't had the time to show you, but we've already scaled this platform down to 48 multi-well, for example, which is already, way more than I was expecting 5 years ago, and we put down papers already on how to do complete in-house full 3D printing of the devices that you might need to do that you might want to use here. So there are ways to improve that aspect, and I think when it comes to having advantages, I still believe that there are lots of examples, including one that I showed you today, but we have at least another 2 examples in which the animal model cannot just be used. You don't have an animal that has the same disease mechanism. The example I show you is a disease which in humans is an autosomal dominant disease. In mouse, you need a knockout. Are you going to really be able to translate any therapeutics using only the animal model, the mouse model? I'll be very surprised, because you just cannot, in that case. And there are multiple other examples like that, or cases in which, like in other neuromuscular disorders, like you can think about the example of my tubular myopathy, where the animal model, the mouse model, just doesn't have the liver phenotype that eventually was found out to be quite prevalent in humans when they had the second hit from the liver toxicity of the adeno-associated viral vector. So, there are cases in which your animal just cannot do the job. And this is where I think it's a great integration to have both things together. You look at the physiology and the metabolism in something that, needs to be an animal, because obviously we all aim to have a quasi micro physiological platform, but at the end of the day, it will always be an approximation. So you will probably need to test some of this in vivo setup anyway. But there are systems and there are target diseases in which the current models have just a number of limitations that you can't just put all your eggs in that basket, I think.
Thank you, Saverio.
Yeah, and I just wanted to highlight that, certainly from the regulatory perspective, if there are no appropriate animal models or relevant ones, clearly, that's where these types of models would potentially be the most useful. And so I don't want to discourage people from using these, as long as you can provide that justification. I think that the reviewers are going to be looking for justification, and they're not going to be as familiar with the models as you are. And so you may need to specify obvious things sometimes in the study report to ensure that people understand the result, they understand the model, understand the basis of that model, what the strengths and limitations are. So in a sense, you have to kind of take the reviewer through and convince them of the fact that this is an important model, because, a priori, they're not going to know that in advance. And so I think that is where it's a communication thing. I really think, they're not unreasonable people. I mean, they're scientists. So you have to kind of convince them with the evidence in a sense. And I think that there's definitely going to be acceptance of that.
And if I can add something else, if you don't mind, because I forgot, there's another relevant point, is that, coming back on the therapy angle rather than the disease model angle, the example that I was making of the AAVs, I think is still a pretty relevant one, because as of today, the vast majority of the knowledge we have around adeno-associated viral vectors and AAV-based gene therapy is on the specificity from animal studies, most of which is mouse. And we've seen the recent examples of the various myotropic AAV, the one muscle specific, that, one single amino acid change made a difference between tropism in non-human primates versus mice. So, having a way even to know from the very beginning, I'm handling a new serotype that actually works on my human target cells in a quasi-physiological setup. I don't think it's something so obvious, and something that it's worth probably investing on, because the other option is that you go a fantastic pipeline with a fantastic result in terms of toxicity and efficacy in a mouse, and then just that vector doesn't hit your cells in humans, and then what do you do with it? So I think it's the integration of the two systems that is gonna be a game changer.
Absolutely. Dr. Engel, would you like to add anything on the how these systems can address such limitations of translating preclinical results into the clinics?
I mean, I absolutely agree that I very much love mouse models, but mouse models have their limitations. And I think as we're learning more and more about human biology, those differences are becoming more pronounced. The other point that was made around modalities, we are certainly moving into a world in which modalities are very much human specific. You know, in the past, you could certainly believe that a small molecule might bind to a protein between mouse and human quite similarly. But now as we moved into modalities that include things like antibodies and nucleic acids, these are going to be very human specific. And as was pointed out, something like the delivery vehicle can be very human specific as well. And so we do need models that are able to capture that. And then what hasn't been mentioned is that, you know, we understand very well that, you know, particularly laboratory mice are a unique entity unto themselves, very homogeneous, and the human population at large is very diverse. And really, these types of systems allow us to do some of that de-risking early on, right? Because again, well, we all, I think, acknowledge that animal models or the use of animals isn't going away. What we do want to do is use them as thoughtfully as we can. And that means anytime we can de-risk something in an in vitro system so that we have higher confidence that when we do have to go to that animal model, that again, we're probably using more for PK/PD than for necessarily an efficacy model, that we are using them as appropriately as possible. And this is particularly true when it gets to things like gene therapies, in which really the species we often have to go into is a non-human primate, because we want to be very thoughtful about that kind of use. So the goal here is to really understand what we're working with so that we put the molecule with the highest probability of success into our animal studies so that we have one successful animal study and we don't have to reiterate through animal studies. And I think that's where there's a tremendous amount of value to the pharmaceutical industry and biotech in order to really drive these things forward. And I completely understand Nikisa's frustration that she's not seeing more of this data coming. I think many companies are investing in this, but you know, it's a learning curve for everybody, right? We have to understand, where do they add value or where do they add unnecessary complexity? One of the things we haven't talked about is the more complex the system is, the more complex the data analysis often is. And so trying to understand, you know, through the noise and through just the volume of data that's produced, what's the meaningful components that come out of it? What's the best way to look at the data? And really make sure that you're understanding the true value of what you have. And so I'm fully on board with, you know, there are some significant places where they're just going to have a huge impact. We are all in the phase of learning together, and it'll start to be more commonplace as we move through.
Wonderful. Well said, Sandy. I appreciate it. Graham, maybe you can say something about, you know, what are the obstacles of creating this pre-competitive space in that specific topic, and then I'll ask Saverio to close with some references on the patient voice in these projects and how you've been powerfully collaborating with those.
Yeah, so I mean, the biggest hurdle that I see in these pre-competitive spaces is data sharing and trying to facilitate the transfer from pharmaceutical to model developer to the regulatory agencies, and trying to, you know, agree on some commonalities where we can share on a level platform is a challenge. And I think that as these consortiums like Magic, I'm very hopeful that you know, you'll be able to share and distribute this data more widely so that we can level set and build upon the successes that we're seeing here for enabling more of these types of collaborations in the field.
Wonderful.
And we already started doing that, Graham. And so we have we have to do it because it's one of the obligations we have. But besides that, we've done it already, and we have, for example, in our case, we have full, extremely long and detailed protocol paper on how to do that. We have all the files to the 3D printing on this, all publicly available. So not the best business model if I was working in the private sector, but as a clinician is what I like. At least, you know, everything is there, and hopefully will be accelerating development of these products. On Ramona's point around patient involvement and involvement of patient advocacy groups, this has been, you know, a part of all this journey from the very beginning. I mean, as you might have noticed from my closing slide, I'm working closely with 6 different charities and patient associations on different projects using this platform. And as an example, I always mention that if it wasn't for a patient advocacy group, specifically, namely Cure CMD, which is they're based in the US. I'm not sure if I would have gone into this path, because it was just by talking with them and say they approached me and they say, you know, one of the former scientific directors approached me and said, look, we got this IPS lines that we generated from 3 patients from our registry. We think that the work that you're doing with IPS is cool and it's promising. That was probably nearly 15 years ago. We just want to send them to you. We think you can do something good with that. And effectively, you know, then when it came to having some preliminary data on the platform, we said, oh, we got already patient-derived material, IPS cells that we can differentiate right away. So, and that helped us having a model within a couple of years. So, you know, it was kind of started from them, and now all the preliminary data that we had from MAGIC, coming from projects that were funded by smaller charities, both in Europe, in the UK, and in the United States, and within MAGIC itself, we have 4 different charities informing both the development of the project and disseminating the finding and targeting both patient communities in Europe, in the UK, and in the United States as well.
Wonderful. Thank you, Saverio. Thank you to the whole panel. I wanted to highlight that really the points raised on data sharing, collaboration across multiple countries, and really having a open mind on how we can best tackle issues in science, because this will really bring us to progress in the future, was really the point of this webinar. And again, I want to thank you, our guest speaker today, and all our panelists. And I will hand it over to Dominic to close out our meeting. Thank you, everyone.
Yes, thanks, Ramona, and thanks for that great presentation. We really thank everyone for attending, and we will be following up with the YouTube link post completion of this webinar. We do have another webinar coming up next month on December 12th at 11 a.m. ET, the power of data sharing and collaboration across LGMDs in a pre-competitive neutral environment. We'll be sending out registration emails for that. We hope you can join us for that as well. And thank you. We have our contact email again at the bottom if there are any other questions. Thanks for joining, and have a great rest of your day.
Thank you.
Thanks again.
Bye, everyone.
Bye-bye. Thank you.
Bye. Thank you.